Thank you. So I'm going to be talking about super resolution, about ways to beat the diffraction limit in fluorescence microscopy, and I'll talk about what are the theoretical, computational, and statistical limitations to how much we can get out of a sample. So I'll start by talking about two things called storm and palm imaging, techniques for beating the diffraction limit. I'll talk about the theory of storm and palm. I'll talk about some ongoing work on image analysis algorithms, and then I'll talk about some theory we've done on optimizing and controlling acquisition. So just to review the diffraction limit, in an ideal world, if we had 2-point probes, a lens would form 2-point images, and we could cleanly see that there are two things there, and we'd see exactly where they are. In the real world, if these things are too close to each other, we get blurs that form. And if they're really close, well, the blurs form in any case, but if they're really close to each other, the blurs will overlap, as shown here in a standard textbook image. And eventually they get so close to each other that you can't see that there are two. And the origin of this effect is quite simple. If we start off with an object and look at the point of focus, the point of focus is a point of constructive interference. All of the different paths that light could take involve the same amount of time by the principle of least time, so everything's arriving in phase. If I want to get a zero of intensity, if I want a point with no light, then I'm going to have to go to a point where the different paths are interfering destructively, which means that the average difference in distance between two light paths is going to have to be about half the wavelength of light. Without that, we're not going to be able to get destructive interference. And so, although no one would actually do the calculation this way, conceptually, if you were to work out what that would require, it would form a zero of intensity at a point in the image plane that corresponds to something this far away in the object plane, about half the wavelength of light. And so what that means is that if you want to see two distinct objects, you're going to have to make sure that the other object is forming an image that's at least this far away from the first one. Otherwise, they're going to overlap each other and you won't be able to cleanly resolve them. So if we look at the effect that this would have on image quality, these are just simulations using realistic point spread functions. Down to about half the wavelength of light, you can cleanly see that there are two distinct objects. Once we get substantially below that, at best you might be able to look at this and say it's asymmetric. There must be some structure down there. In the past several years, a number of tricks have been developed to get around this. I'm going to mostly talk about things related to the top row of images. On the left, we have microtubules imaged with conventional fluorescence microscopy. On the right, we have microtubules imaged with a trick called palm or storm. And you can cleanly resolve individual tubules here, whereas on the left in the conventional image, you're just guessing whether there are multiple tubules. If we were to look here, we could probably guess that there's more than one. We would not be able to resolve this detail. Now, this family of techniques that I'm going to talk about goes by a number of names because it was invented by several different people around the same time. PALM is photoactivation localization microscopy. STORM is stochastic reconstruction microscopy. There's also Palmyra, FPalm, IPalm, and probably there's D-storm, DAO storm, but they all work off of the same basic concept, which is that if we had two things that were both fluorescing at the same time, they would form an overlap image. But what if only one of the molecules is fluorescing? If only one of them is fluorescing, this blur might have a size comparable to the wavelength of light, but the blur is a shape, and we can find the centers of shapes. That's not a problem, at least if the shape is well-defined and not too noisy. So I could say, all right, there's a molecule there at a point corresponding to the center of the blur. Later on, a different molecule is fluorescing. And I can again look at the center of that shape and figure out where that molecule is. And if we do this enough time, we can build up an image with quantitative detail in it, with quantitatively valid reconstructions of the molecular positions. The basic time course of an experiment, you can imagine that all of the black dots are molecules. And I have drawn circles around the ones that happen to be fluorescing. The radii of those circles are about the wavelength of light. It's showing the size of the blur they would form. And I've drawn these lines just to indicate that we could localize anything that's on. And initially, by chance, some random subset of them are on. And if only a small fraction of them are on, the odds of an overlap are low. Now, we don't get to determine which ones are on. If we could determine which ones were on, then we would already have a level of information and control that would enable us to do a lot more things than just take pictures. Later on, by chance, some different subset are on, and again, we find all those positions. Every now and then, by chance, two blurs will overlap. And the overlap of those blurs is an important topic that's going to be a big feature of this talk. Because when the blurs overlap, unless we are careful with how we process this image, we would just look and say, oh, well, there's something there at the halfway point between them, at the center of this blur. And that, of course, is junk information. There's nothing that's actually right here. There's something here and here, but not there. And so we have to think about what can we do to minimize the effect of those overlaps. The basic idea in most of the systems that are understood is that you have three sets of states. We have a dark state. This is our default state. It doesn't fluoresce. But if you hit it with the right wavelength of light, it activates. Now it goes to a metastable state. And that metastable state isn't going to emit a photon and return to the dark state anytime soon. But if we hit it with another wavelength, it could go up to a band of excited states separated by vibrational transitions, and then it could fluoresce. We could hit it again, and it'll go up, and it'll fluoresce. And it'll do this a number of times. Each time it fluoresces, it gives us another photon. The rate at which it fluoresces is proportional to the intensity of the green beam. The chance of returning to the dark state, or the rate at which it will eventually return to the dark state, is also proportional, at least in many experiments, to this green beam. And so when these two rates are both proportional to the same thing, their ratio will determine the number of photons you get. And so in many cases, the number of photons you get is actually invariant. Now, all of this is heavily caveated because these photoswitchable molecules, as they're called, are an area of very active investigation. What I'm describing is a typical mechanism, and there are still a lot of questions. But this is the basic idea that works well enough to understand most experiments. Now, given that we can switch things on and off, how can we localize them? I've talked about finding their shape, finding the center, but I want to emphasize how much information we can pull out. This is a simulated image. And if it's perfectly centered on a pixel, we get a symmetric image. If we shifted a quarter pixel, you can see that the image has changed. And anytime there's something apparent to the eye, there's probably a way for a computer to extract that. And if we shift it by another quarter pixel, now again we get some symmetry in it, but now the symmetry is about the pixel edge rather than the pixel center. So the key thing to take away is that even though the width of the point spread function is very large, small shifts, even subpixel shifts, can be detected. The problem is noise. That simulation did not have any noise. In the real world, photons arrive one at a time, and the sequence is not predictable. So here's a simulated image with 50 photons. Your guess is as good as mine where the molecule actually is. 200 photons. I'm pretty sure it's not around the edges. I wouldn't want to place a bet on which of these 6 pixels it's on. 800 photons, it's pretty clearly in the center, but again, due to some of the asymmetry in the image, your guess is as good as mine what the subpixel shift might be. 2,500 photons, it's fairly symmetric. I could say that it's probably pretty close to the center. And the precision with which we could do this quantitatively is approximately the wavelength of light over the square root of the number of photons collected. Now, when I say the wavelength of light, I'm assuming that we have a diffraction-limited image. But all of these tricks are really not beating the diffraction limit. They're beating the point spread function limit. If you were imaging through something with significant scattering or aberration, then all you have to do is replace some measure of the point spread function width there, and to within some factor of 2 or pi, the same limit will again apply as long as your background is low. And I emphasize that to this audience because I have seen some work on doing super resolution with blinking autofluorescence. And so with Now when we start talking about blinking autofluorescence, you don't have to think about very specific labels. You think about things that might be going on in an organism. So think of this as beating the point spread function limit, not as beating the diffraction limit. Just to show you some more pictures of what is possible when you work at this high resolution, the top row are images of microtubules from Xiaowei Zhuang's group at Harvard, and these were taken with conventional microscopy. These two boxes are zooming in on these two boxes on the left. The bottom row is the same thing, only they've done this reconstruction on the images, imaging them one at a time. And you can see single tubules. If you wanted to test some theory about the organization of the cytoskeleton, of how some drug affects the cytoskeleton, of how cytoskeletal organization is involved in some dynamic process in the cell, now you have the information to start talking about these questions with direct images. Just some more picture. This is with two different labels, microtubules and mitochondria. You can see what's on one side of a membrane and what is on the other. This was done in 3D, again in Xiaowei's lab. This is not the only way that people have found to beat the diffraction limit. People were taking electron microscope images for decades, but those don't play so nicely with live cells. It's non-invasive, unlike near-field techniques. With FRET, you can certainly use an optical probe to obtain information on nanometer scales, but you're generally working with pairs of probes. Here, you're working with the entire structure. It's usually a simpler optical setup than stimulated emission depletion. It usually works with lower excitation powers than stimulated emission depletion. And although negative index materials get a lot of attention for being the diffraction limit via a completely different route, the materials for these setups actually exist in a mature form. Now, I'm a theoretician, so what am I doing talking about this? So this is from the Zeiss company website. Ernst Abbey was one of the founding participants in that organization, and they just talk about how prior to Abbey's work on formulating a theory of imaging, a lot of microscope design had to be done on a trial and error basis, but once there was a mathematical basis for making predictions, refinements could be done much more rapidly. Well, now that Abbey's limit doesn't apply, now that people have found ways to circumvent that and pull out information beyond it, the question I work on is what limit replaces it? What theory do we need to guide development? So a problem that has occupied a lot of my attention in this field is a very simple one. Are there two molecules in this image? Because everything that I described only works as long as you know that the bright spots you're seeing are single molecule. As soon as they are multi-molecule, now you have a problem. If you just try to localize that, you're going to get junk out. And so you can work at a very low activation fraction. You can tune your laser beam such that the odds of overlaps are so small that you don't need to worry about it. But what if you want to work quickly? What if you want to get as many of these as possible? Or what if things are aggregating during the time course of an experiment? This matters. And so we could look at this and say it's asymmetric. There must be two in there. But the question is, how do we teach a computer to recognize that, and what are the implications if the computer gets it wrong? So the theory that I formulated assumes that we do all of our work on a single diffraction limited spot. Again, replace lambda with the PSF width if you're working in some other regime. I'm going to assume that there are n molecules per spot, and the one lever that we can tune in an experiment is the activation probability per molecule. I don't get to decide which ones are on, but I get to play with the intensity of the laser beam that switches things between activated and dark states. So I get to decide what fraction of them are on. And as long as we're outside of the FRET regime, as long as we're outside of a regime where the molecules interact with each other, the probability of getting m molecules on in this spot is given by the binomial distribution. And we want to pick our activation probability such that P1 is large. We want lots of single molecule images. We certainly don't want zero molecule images. Those have no information. And for m bigger than 1, we don't want a high probability. Now we can work out a minimum number of detection cycles from this. We could just say that the number of cycles we need is going to be proportional to the number of molecules divided by the single molecule detection, the probability of getting a single molecule image. And what's interesting is that there is a minimum number of cycles. If you set your activation probability too low, not surprisingly, you're getting a lot of zero molecule images. If you set it too high, you actually introduce more junk than good information. That's not too surprising. Now what we can do is we can calculate the rate of overlap errors. So I'll define that as the probability of a multimolecule image divided by the probability of a single molecule image. And if I worked at this limit, it's 0.7. For every 10 good images, there are 7 bad ones. So we could turn down our activation probability, reduce the number of multimolecule images, get a slow experiment, or reject the overlaps. Now we get to do a fast experiment. If you read the literature in this field, everybody has their own homebrew image processing techniques. Some people are starting to adopt others, but there are still a lot of homebrew things. There are things that have been borrowed from other tasks, algorithms that were good for the tasks that they were designed for and are sometimes very good for this task, sometimes not. Rather than trying to characterize each individual algorithm, I'm just going to assume that the algorithm is characterized by a set of probabilities. Right? When it gets this 2-molecule image, there is some probability that it will get it right and realize there are 2 molecules, and there's some probability that it will get it wrong and record a position from that image. And we want F1, the probability of accepting a single molecule image, to be high, and we want all of the other ones to be low. Now, there are many different ways to do this. You could generally categorize them into shape tests or intensity tests. A shape test, you just look and see whether it's an ellipse. Lots of ways to do that. Intensity tests, you just say, well, if there are a whole bunch of photons coming from this, there's probably more than 1 molecule emitting. That's a pretty good rule, except that the photon counts are exponentially distributed in most experiments. So something that's twice the average photon count might still be a single molecule image. And we'll get to some of the math of that in a little bit. We can now calculate the error rate, and so we have to readjust our calculation. We have to include the probability of getting an M-molecule image and the probability of accepting it. In most of my work, I focus on the 2-molecule events, and I'll give you a few justifications. One is that as long as you're working below the maximum activation probability, this is more common than this, and you can show that mathematically. Another justification is that this elliptical image might be hard to reject. Imagine if they're very close to each other. It's only slightly elliptical. Good luck distinguishing that slight ellipticity from noise distortion. Whereas this thing is large, it may be irregularly shaped, and although photon counts aren't a perfect proxy for number of molecules, they aren't a bad proxy. If you have 10 times the number of molecules and it's exponentially distributed, the odds of that, 10 times the average number of photons and it's exponentially distributed, the odds of it being a single molecule image are very low. We can work with the 2-molecule error rate because if we were to calculate the total error rate, if we were to include all of the 3, 4, 5, and 6 molecule images and so forth, we could make the assumption that the probability of accepting a 3-molecule image is no higher than the probability of accepting a 2-molecule for the reasons I just gave. So we could establish an upper bound on the true error rate based on the 2-molecule error rate. And that upper bound is never more than 40% above. So you're close, and you're close in a predictable way if you focus on the 2-molecule error rate, which is what I do in most of my work. We can work out the existence of a maximum error rate. And basically, that is just the ratio of the probability of accepting a 2-molecule image to the probability of accepting a 1-molecule image. This scaling is not so surprising, but an expected result isn't so bad as long as some of the other results are a little bit non-trivial. If we were to take this relationship between activation probability, number of molecules, and error rate, and plug that into our formula for probability, we could work out the minimum number of detection cycles as a function of error rate and algorithm performance. This result is a little bit non-trivial, especially the exponential factor. But what's key is that you can see that it saturates. As we turn up our error rate, as we decide to accept more and more multimolecule images, not surprisingly, there are diminishing returns on time. We can quantify that. And so we can say whether you're making a good call or a bad call by turning up your error rate. Now, some people, there's increasing work on this. Some people say, I'm going to work with the multi-molecule images. There are ways to do that. You could fit a multiple molecule model to it. That's a good way. You could work on different ways of representing the information in an image and compare that to different ways of representing a multi-molecule image. I know there's a little bit of preliminary work on that. There are some bad ways, like fitting one at a time and subtracting things out. But again, I don't want to get into the weeds of specific methods. What I just want to say is, let's work out the maximum improvement you could get if you use these techniques. Let's say we've got some algorithm that can work with the multi-molecule images. And it works perfectly up to some number of molecules. If it gets a 2-molecule image, it knows it has a 2-molecule image. And it finds those 2 positions perfectly. But eventually, there's some limit. It can't fit some horrendous number of molecules. I'm using 3 for simplicity. And when it gets something it can't handle, it knows it can't handle that. So it says, I'm not even going to try to analyze this. It's as perfect as you're going to get from an algorithm. And the question I'm going to ask is, by turning up your activation probability to bring in more of these multi-molecule images and use them, how much time do you buy? So this table shows the number of molecules that the algorithm can discern and some measure of the minimum number of cycles needed. And 1, 2, 3, 4, 5, you go down by a little bit less than an order of magnitude. When you go from 5 to 10, you actually buy a pretty substantial improvement. And but all of this is under ideal assumptions. I have seen published work on 5. I believe that there are people trying to get up to 10. And this is very idealized, but at least we can quantify what is the maximum that we could hope to get. And so when someone develops a technique that works with up to 5-molecule images, you can then ask how much time, how many cycles did they need to reconstruct the image, and how does that compare to the theoretical limit? So at least we have a benchmark. Now let's talk more about the implications of these multi-molecule images. If I've got 2 molecules right next to each other, and I image them a whole bunch of times, sometimes the one on the right is activated, sometimes the one on the left, sometimes they're both. And if I were to just make a map of the position estimates, I'm going to get 3 little circles, which I've done in different colors only to distinguish them. And what you would want is that the circle in the middle, which is clouded out just like the rest of them because these estimates have some statistical variation, they have a variance. If you want to cleanly resolve the left molecule from the right molecule, the one in the center had better not stand out. There had better be very few images in this overlap spot. And so what we need to do is we need to pick a 2-molecule error rate such that there are very few in here and most in here and here. Now, if we think about what an algorithm does when it does a shape test, if they're right on top of each other, it's highly elliptical. The acceptance probability is high. It's not going to be able to tell that only slightly eccentric ellipse from a circle. Whereas when they get farther away, now it's a very eccentric ellipse. And in this very eccentric ellipse, you really ought to be able to tell that that is a 2-molecule image. I'm not going to assume that the acceptance probability is 1 at 0 separation. I'm going to allow for the possibility that the photon count test gives at least some useful information, but I assume it saturates, I assume it cuts off, and we have started to do some benchmarking of several different algorithms, and indeed, they follow a curve more or less like this. So your 2-molecule signal is going to come mostly from things that are closer than this characteristic length scale. So we can calculate as a function of algorithm performance and activation probability basically how many counts you're getting from the 2-molecule images here and how many you're getting from the 2 on the left and right. And we can get a criterion. If we want the single molecule spots to be distinguishable from the multi-molecule spots in between them, then that length, that measure of how far the molecules have to be before the algorithm can discern them, should be less than this. Less than something that depends on the wavelength of light, your target error rate, and 2 measures of algorithm performance. And the key thing to know is that F2 of 0 is in the denominator. All right? F2 of 0 is the probability of accepting an image if they're practically on top of each other. And the only way that you can do anything then is if you work with the photon count test. And what's interesting is that, first of all, this is going to be less than 1, which means that you've just improved your tolerances on L. So if we do a photon count test, which is computationally trivial, we improve our tolerance on L. We can work with a larger L. That's a dumber algorithm, an algorithm that doesn't necessarily require as many iterations or as good of a signal to noise. So what we're learning here is that the intuition that you should use a photon count test is right. It actually produces a quantitative improvement in the quality of the reconstructed image. Now, we have started benchmarking several algorithms. I'm going to show one of them right now. This was done by an alum of my lab who is actually here today and is now working in Dr. Stephen Small's lab in neurobiology, Forrest Hippensteel. And what Xiaowei Zhuang's group does, they came up with this approach. They fit these spots to an elliptical Gaussian. And so these 3 parameters in the exponent are fit parameters. And this would be an example of a highly elliptical Gaussian. You would definitely have A and B different from each other. This would be an example of 2 molecules on top of each other. A and B are exactly the same. They can infer semi-major and semi-minor axes of the ellipse from these 3 fit parameters. And if the difference is more than 15% of the average, they reject. They take that as an indication of ellipticity that works for their signal to noise. We feed in simulated images. And we don't just do that because I'm a theorist and that's what theorists do. We do it because the whole idea of an image processing algorithm is to pull something out. All right? But if you're going to compare, you already have to know what's in there. And there are good analogs of a phantom for super resolution, DNA rulers. But to acquire a very large set that uniformly samples parameter space is hard. Fortunately, there are realistic point spread function models, realistic noise models. So we just feed in a whole bunch of simulated images with reasonable pixel sizes, reasonable signal to noise. And then we see what comes out. We sample parameter space uniformly and see what the 2-molecule acceptance probability is as a function of molecular separation. We did this for a few different pixel sizes. Background counts per pixel are scaled to pixel size. A few different photon counts. Some people look at these numbers and say that's absurdly high. Some say it's absurdly low. It really depends on your system and your fluorophore. What I want to draw your attention to is that it doesn't go to 0. Any guesses why it doesn't go to 0? Well, I said that the photon counts are a random variable. They're an exponential random variable. That exponential distribution is peaked at 0 photon counts. So some of the things that we're calling a 2-molecule image look like this. This is, we would expect to be elliptical. Some of them look like this. You can barely tell that there is a second molecule in this image. So what harm are we doing if we accept this? Accepting this is clearly harmful. You're going to get an estimate there. Accepting this, maybe it won't bias your estimate at all. So what we did was we started removing different things from the simulation. We went back into that same simulation and said, let's pull out all of the 2-molecule images that have a really high contrast. Then we'll pull out all of the ones that have a contrast still high, but relax the criterion. So we toss out more. And then we toss out more. And we ask, how many do we have to toss out before it's not accepting anything? And the answer is that we have to toss out everything with a contrast bigger than 4:1 before we get 0 acceptance probability at these large separations, greater than about quarter wavelength. So what this is telling us is that as long as your images are, have a contrast above that, you won't get, it will be accepted. And now the question we have to ask, a question we're working on, is what does that mean for localization precision? Are those high contrast images returning bad position estimates or good position estimates? And that's something we're working on. We also looked at the single molecule acceptance probability. We just vary photon count. We play with background. And not too surprisingly, you get an acceptance probability above 90% as long as you have more than about 600 photons per molecule for a substantial range of backgrounds. And this is not too surprising. This is also good news because there are a lot more photoswitchable molecules with 3-digit photon counts on average than 4-digit photon counts on average. Now, what if we were to work at the high end of the distribution? Before I was talking about the very high contrast images where one of the molecules is producing 2 photons and the other is producing 2,000. But what if we toss out anything at the high end where the total photon count is greater than alpha times the mean? So we played with that, and we plot F2 of 0 on the vertical, the fraction of 2-molecule images that we would accept by this method, F1 on the horizontal, the fraction of 1-molecule images. And we could get 95% of all single-molecule images while tossing out more than 20% of 2-molecule images just by having a criterion of 3. This buys us some leniency on the tolerances for our shape test and speeds up the processing. Now, that's only half of the game. Figuring out whether it's 1 molecule or 2 molecule is the beginning of the process. Then you have to ask, given that we've accepted this image, where is it? Where is the molecule? And there are a number of different approaches for that. There is a statistical theory, though, showing that the maximum likelihood estimate has the lowest variance. If you were to image the same thing again and again, you would get images that differ every time only because of noise. If you were to feed in that set of images and estimate the position as a parameter, and then look at the standard deviation of that set of estimates, that is minimized when you use something called maximum likelihood. And the basic idea is you take your image and you take a model for the noise. So you have to know something about the noise characteristics of your system, and you have to have a reasonably good point spread function model. And then you calculate the likelihood of getting that data given those parameters. And you play with the parameters until you've maximized the likelihood of your data. It's used in a lot of different areas of science for data analysis. It's different from the least squares criterion because it requires a more detailed model, but if you have it, it gives a lower variance. What we recognize is that this can be sped up because most people do not fit to the exact point spread function, largely because the exact point spread function is not always known. You can have a fairly good model. That Airy function is a good approximation, but as soon as you include apodization, as soon as you include reflectance, as soon as you include anything else that's going on in your imaging system, it's only approximate. So for analytical simplicity, people fit to a 2D Gaussian. Well, this function is separable, which means that if we were to sum all of the intensities in this column, we would get something that only depends on the x-coordinate of the molecule. And if we sum in this row, we get something that only depends on the y-coordinate of the molecule. So if you have an L by L data set, we could do our fits on L data points instead of L squared data points. This is a square root speedup. And we looked around in the literature. Strangely enough, no one has used it. And so our first thought was, ooh, maybe it doesn't work. Maybe there's a nasty thing we're overlooking. The one approximation that could break down here is that your region of interest is not infinite. Some photons fall outside this area, and some fall in gaps between pixels. And so we asked the question, what happens if a significant amount of photons are falling outside your region of interest because it's small or because you drew your ROI off center? What happens if you have a lot of dead space between pixels? So we just fed in a bunch of images generated with realistic noise and point spread functions, fit them to this approximate point spread function using this assumption that we only need rows and columns. And what I want to draw your attention to in this complicated plot is that on the vertical scale, we have the localization precision. On the horizontal scale, we have the position of the molecule relative to the center of your region of interest. This black curve with diamond points on it is the theoretical minimum. There's an established theory for it. There's software developed by Raymond Ober at UT Dallas showing that you can use to calculate the minimum standard deviation. And then we did this in a very ideal case. Large region of interest, no dead space between pixels. Here we do non-ideal cases. We increase the dead space between pixels. We decrease the size of the region of interest. And what we find is that unless you're a couple pixels off from the center of the region of interest, you are within about 20% of the theoretical minimum, often less than 20% of the theoretical minimum, while speeding up your data analysis substantially. It does go up substantially here when you're off-center, but we made one ideal assumption here, 0 background. As soon as we add background, now there's no sharp rise. The overall uncertainty has gone up, but it's gone up for the ideal case as well as the non-ideal cases because that's what background does. The reason why it doesn't go up at large separations, at large distances from the center when you have background, is because model mismatch only really matters at the tails. At the tails, it matters whether something is dying off exponentially or power law, and that makes a big difference for your maximum likelihood estimate. As soon as you add background at the tails, that becomes a lot less important. And so what's interesting is that this algorithm actually gets better while being faster as soon as you add a realistic issue, background. Here is another test of the localization precision. We played with the photons per molecule, and we generated 10,000 images at each subpixel position. And what we find is that the circles have radii that's twice the standard deviation of the estimates. Every time we multiply the number of photons by 4, the number of circles that we can fit in approximately doubles. That just means that the resolution is going as 1 over the square root of the number of photons. This is entirely expected by theory. It's a good sign. There's no bias. There's no sensitivity to pixelation effects. We have run other algorithms, and I won't go into what we have found, but we have seen algorithms that are very sensitive to pixelation. So this is another good sign. Now, the final question that I want to tell you a little bit about is what happens when we bring in bleaching. Everything that I've been talking about so far ignores the fact that eventually a molecule is irreversibly photodamaged. And so I made these predictions for the minimum number of image acquisition cycles, assuming that the molecule will still be there. If you don't get an image now, maybe it gets activated with a neighbor, so you have to toss it out. You can activate it again. This is our prediction in the case of bleaching. And this T is the average number of images per molecule that we want. What if we can't get that because our bleaching rate is greater than 1 over the number of cycles times the duration of a cycle? So we need to model the population. So initially we assume a very simple 3-level or 3-band model as before. And we assume that bleaching happens from the excited state. There's still a lot of research on bleaching, but this seems to be a good assumption as far as we can tell. And we get that the rate of change of the number of molecules present depends on the bleaching rate and the activation probability. And we can relate all of this to the error rate. Now you're wondering, why does algorithm performance come in here? That's because this error rate is a measure of the quality of the reconstructed image. And to work backwards from that to an activation probability, you have to factor in the algorithm performance. The one thing we can control is the activation probability. Through that, we control n dot. And what we want to do is we want to optimize an integral of single molecule image probability. This depends on this is the probability that one of the n molecules is on and the others are off. That depends on the probability per molecule, which depends on n dot. So now we've got this integral that we have to optimize. It depends on the derivative of a function. And I've spent a lot of time teaching classical mechanics lately. The one thing you learn in classical mechanics is the Euler-Lagrange equations. So we have a Lagrangian problem here. This is our Lagrangian. And it only depends on the derivative of a function, not the function itself, which means that we have a conserved momentum here. And if we have a conserved momentum, that means that n dot is constant. So n of t decreases linearly in time. So what I'm plotting here is n of t for two different error rates. These are constant error rate schemes. I plot the corresponding activation probabilities as a function of time. The one question we could ask is that we've only made that integral stationary. We've basically taken the first derivative of that integral and set it equal to 0. We haven't proved if it's a maximum or minimum. Now, there are very few, it's hard to formulate the necessary conditions for making an integral a maximum or minimum. However, sufficient conditions are known, and it just so happens that our integrand satisfies them. T1, the total amount of time that you only get one molecule on, is a maximum. What about the amount of time that I get 2 molecules on, or 3 molecules? Well, for 0, 3, 4, and 5, they're all minima. So that's good news. We've minimized the number of 0 molecule images in this constant error rate scheme. We've also minimized the 3s, 4s, and 5s. The 2 is special. For small error rates, T2 is a minimum. For larger error rates, it's a maximum. I maintain that you actually want to work at the larger error rates, and here's why. Because nothing's perfect. So what happens if we deviate from a scheme that is maximizing T2? When we deviate from it, the number of 2 molecule images will go down. We've also maximized T1, so the number of 1 molecule images will go down. So it's compensated. Now you might be saying, wait, aren't high error rates bad? Well, sure, but those error rates are constrained by the performance of your algorithms. So if you have a good algorithm, and then you work in a scheme that is supposed to give a constant error rate, anytime there's a deviation, the number of single molecule images will go down, the number of 2 molecule images will go down. So it's robust. Now, people have done some single wavelength schemes. I'm going to tell you a little bit about them. In a single wavelength scheme, the wavelength that activates is also the wavelength that causes fluorescence, all right? Which means that you now only have 1 knob to turn, not 2. There are a lot of different models for how this might work. The dynamics of these states are still very much debated. So rather than presenting you with the grand unified model of single wavelength super resolution, we analyzed 4 models, all right? Each corresponding to plausible mechanisms that might be supported by data that are supported by at least some data in the literature, but are still subject to controversy. This one is very similar to everything I've been telling you about, all right? It's just that the molecule that sends it from the dark to the activated, the wavelength that sends it from dark to activated is the same that sends it from activated to excited. But a lot of schemes work with a triplet state. So the wavelength that can send it from a default activated state to an excited singlet state and cause fluorescence can also send it to a dark, long-lived triplet state from which it takes a while to return. Our theoretical approach is more complicated. Suffice it to say that you have to look at the bleaching rate per molecule. That is the parameter that all of our models depend on. In the first case, we get something that looks like this for the number of molecules. They no longer decay linearly. They decay as basically a square root function, which means you have to play with your activation probability versus time, and you're no longer working at constant decay, but you still, interestingly, have a constant error rate, right? And it's robust now for small error rates. Different integrands we're playing with, robust for small error rates. Another case, much more complicated. We go from activated to triplet, stays there for a while. Bleaching happens from the triplet state with the absorption of a second photon. There is some data for this in at least some cases. Different time dependence. You're going to have to play with your activation probability differently. It's robust at high error rates. Complete opposite of what we had here. Another mechanism. This time your error rate is not constant. The intensity of your laser gets tuned in a very much different way. In this case, you either get photoinduced bleaching from the activated state, or bleaching without the absorption of a second photon from the triplet state. And if you take nothing else away, you should note that the schemes on each of these three slides were completely different based on a different model of the photophysics, which means that because we only have one knob to turn, when we're turning that knob, everything changes at once. You don't get to compensate for ignorance of one thing by turning a different knob and kind of playing with it by trial and error. You need to know exactly what's going on. So the key lesson here is three different plausible models of the activation and bleaching process, three completely different control schemes with different robustness properties. My message to the people doing single wavelength super resolution is that we really, really need to know the photophysics of the specific dyes being used if we're going to get away with trying to optimize those experiments. Whereas in the two wavelength schemes, you have enough robustness that you don't need to worry. Just really quick, I want to say that there are other things we can understand in this formalism. I developed all of this terminology. I defined all of these variables. We'd like to be able to calculate as many things as we can so that what we have is a tool set that anyone working in this field could use. One thing we decided to calculate was the probability of never getting useful info from something, either because we mistakenly accept a multi-molecule image or because we fail to accept a single molecule image, and these mistakes happen again and again until it bleaches. Suffice it to say, we get something that depends on the bleaching probability per cycle, the error rate, and the algorithm performance. So we have a theoretical toolkit. This is a plot of the probability of losing information versus error rate. I want to leave you with a look at something else in the field and a question. Different techniques, stimulated emission depletion, also beats the diffraction limit. Totally different idea, though. They focus a pulse and raise a bunch of things to the excited state within a region of size lambda. Then they have a second pulse. That second pulse is a donut mode. It's a TEM10, right, or a superposition of TEM10 and 0, 1. No intensity in the center. Strong intensity off center. It's tuned to the downward transition, so there's stimulated emission to go to ground. If you wait, you'll notice that these did not go back to ground because there's zero intensity there. They will eventually fluoresce later, and so you know that any fluorescence detected later must have been from this tiny region. Nowadays they have a CW version of it, but this is the concept. They get beautiful pictures. These are pictures of mitochondria. They stained for two different proteins. You can see inside and outside. What's interesting is that your resolution scaling is 1 over the square root of the intensity, and the mechanism is the parabolic beam profile. In STORM, your resolution scaling is 1 over the square root of photon number, something that's proportional to an intensity. Totally different mechanism, shot noise. I included differential confocal here. It's also based on shot noise, but it has nothing to do with activation. It's been used to do some surface profiling at very high intensities, and they combine a number of different measurements rather than taking a single image. And again, they get 1 over square root of photon count due to a shot noise mechanism, but a totally different process. And so again and again, and there have been other things I've looked at, people get this 1 over the square root of something related to photon count. Is that a universal limit? If they all had the same mechanism, it would be obvious. But there are different mechanisms. And so a question that we're working on, a question I always love to discuss, and I hope to discuss with a few people today, is this universal? Is there either a way to show that when you combine these different steps, you always get the same limit? Or is there a way to combine them and get something better? Because if there's a way to get something better, then there's a gold mine to be dug. If this is the limit, then we know where to go for future improvements. So with that, I'll say that in conclusion, subwavelength imaging is partially constrained by the performance of your analysis algorithms. We're looking at more of an information limit than an instrument limit. We've proven the existence of an optimal scheme for 2 wavelength control. We've shown its robustness. We've proven that single wavelength control requires much better photophysics knowledge. And we have shown that square root speedups are possible in image analysis. I'll thank my sponsors, the Research Corporation, CCRA, it's a grant that the campus has that supports undergraduate research, C-Superb, the CSU program for education and research in biotechnology. On the algorithm side, Shane Stahlheber currently working with me. Forrest Hippensteel used to be working with me. Edward Shore looked at how algorithm performance affects the optimal two-wavelength scheme. For the square root speedup, Rebecca Starr has done most of that work. Earlier versions of that algorithm were studied by Adeline Cornelis and Nahom Yerga. Some work that we have done on stimulated emission depletion was done by Michael Mansell and Triet Nguyen, and thank you for your attention.